put in a lot of service in the university. Presently, is working as professor in the Department of Biotechnology, Kuvempu University. He has total experience of 32 years, and as professor as such, he has 16 years of experience, and he has worked as Dean of Science and Technology. He is director for CDC and IQAC. He has been member of syndicate, member of academic council, director for statutes and governing council, and he has been chairman for Department of Biotechnology Board of Studies, director for School of Biological Sciences. For more than 10 years, he has served as Board of Studies and Board of Appointment, as well as for Kuvempu University, Tumkur University, Karnataka University, Bangalore University, Gulbarga University, and also Mysore University. He has been Board of Examination for all PhD examinations for past 8 years, and also for the PG Board. And he is coordinator for commercial plant tissue culture units. And he has worked for convener for SCST cell and convener for extracurricular activities also. For his credits, he has written 4 books, 205 research papers has been published by him. He has a total citation of Google Scholar around 7,057 with H index of 30 and I-10 index of 106. And in Scopus, it is 4127, and in H index around 23, and RG score of 3,134. His impact factor is around 0.1.73 to 3.43. And he has been PI for 9 major projects, which totally amounts around more than 5 crores of rupees. And in addition, he has guided 48 PhD candidates and 5 MPhil candidates. And he has presented 31 research papers in both international and national conferences. He has 13 awards received till date for his credit. And he has also worked in the technology transfer from lab to land in production of Panama disease-resistant tissue, cultured banana, plantlets, and distribution of plant to the farmers of Karnataka. With this brief introduction, I welcome Professor Krishna, sir, to this session. Thank you.
Thank you, madam. Thank you. Respected professors. Hello.
Yes, sir.
Yes, sir.
Good
morning,
sir.
Good
morning.
Good morning. Good morning, sir.
Good morning.
Good morning. Okay, sir. Can we start, sir?
Yes, sir. You can start.
Yes, sir.
Okay. Thank you. Thank you, madam. Thank you. Today, we'll discuss on ex-situ conservation and sustainable use of threatened medicinal plants of the Western Ghats. Friends, as you know, the plants are the vital for the existence of life in the university. Throughout the ages, the man depend upon plants for his food, shelter, clothes, and also the medicines. So the need of the trace of development of herbal drugs starts from the ancient times. To cure his wounds and other ailments, it depend upon the plant products and also the natural resources. So as you know, India is endowed with the vast resource of the medicinal and aromatic plants. And these plants are used in the traditional Indian system of medicines to cure various type of the ailments. As you know, the before we consider Hippocrates as the father of the medicine. But before Hippocrates, many of our Indian scholars worked and they proved the medicinal property of the plants.
Professor.
Yes, sir.
D. Krishna, sir.
Yes. Yes.
No, sir. Sorry, sir. Please continue.
Yeah. If anything, please interfere also, sir. Any doubts, we will clarify at that time. Okay. So starting from the ancient times, man depend upon the plant and its resources and on the plant products. Hippocrates considered as the father of the that is all the medical sciences, including the allopathic medical sciences, and also he laid the foundation for the Unani system of the medicine. Before Hippocrates, many of our Indian scholars like Charaka Maharshi and Sushruta Maharshi, and even we consider Dhanvantari as the father of the medicine in Indian culture, in Indian system of the medicine, that is in Ayurveda. And we also know that according to the ancient writings, the Vagbhata Maharshi, that is he wrote the 8 disciplines of Ayurveda, that is Ashtanga Hridaya Samhita. These that is traditional system of medicines, Ayurveda, Yoga, Unani, and Siddha. In these systems, they consider that the power, that is the power and the matters, they play an important role in curing many kind of all kinds of the ailments. And before the entry of the Aryans in southern India, that is the first culture civilization starts from southern India, that is the Tamarapani civilization, where the Kaveri and Kannika river joins, and in that region the Tamarapani civilization starts, and where the Agastya Maharshi, and he laid the foundation for the Siddha system of the medicine, and his 18 students, they are called as the 18 Siddhas. They wrote about the different disciplines of the Siddha system of medicine. They postulate that the power and the matter, they play an important role. The power is the Shakti, and the matter is the Shiva, according to the Siddha system of medicine, where the Shakti and Shiva, they play an important role in the genesis of many kind of the things. They also consider that the body, including the cell, even a single cell, considered as is under the control of that is the power and the Shakti. So they state that the power without there is no genesis of the power without the matter, and without power the matter is inert. That's why even in the many of the Indian scholars postulate that without Shiva there is no Shakti, there is no Shakti. Without Shakti there is the Shiva is also inactive, like that. So here, in all system of medicine, either in the Ayurveda and Siddha, and also in case of the Unani system of medicine, where the humors develop in the body, and the entire cell, even in a single cell, and the body or organs of the body is under the control of the powers, that is the Panchabhutas. They consider as the power of main powers, that is Agni, Jala, Vayu, Akasham, Earth. These are the Panchabhutas. These Panchabhutas interacted in the, yes sir?
Sir, only one, that is second slide is visible, sir, to all of us actually.
Yeah, only second slide, yeah. We'll go to the next. We'll go next. Yeah.
Okay, sir.
So these Panchabhutas play an important role in the in controlling of the body. If these Panchabhutas are interacted in different patterns, so development of the humor takes place, that is Vata, Pitta, and Kapha. So the medicinal plants, they play an important role in the curing of many kind of the ailments. So the plants, many of the plants are used to cure various kind of the ailments. Our Western Ghats is considered as one of the hot spots of the world. So it is a mountain ranges, starts from the western side of the India and also known as the Western Ghats. It runs from the western edge of the Deccan Plateau and hence in Arabian Sea. This Western Ghat ranges consists of 1,600 kilometers and it runs across the states of the Maharashtra, Goa, Karnataka, Tamil Nadu, and Kerala, and ended in Tamil Nadu, in Kanyakumari. These are the important peaks in the Western Ghats, that is Anamudi Hill Ranges, that is it is around 2695 that is the meters, whereas it is started by from Misapulimala in Kerala and Doddabetta of the Ooty, Kodaikanal and Chambar Peak, whereas the Mullayanagiri of Karnataka, that is in 930 meters. Then it is followed by Baba Budangiri, Kuduremukh, then Agasthimalai again in Kerala, then Biligiri Rangana Hill Ranges, then Kumara Parvata, Pushpagiri, like that. So Kodachadri is that is 1343 meters. So these Western Ghats hill ranges harbors more than 4,500 species of the plants. Among them, more than 1,200 species of the plants are endemic to this region. They are present only in these regions. That's why it is called as the hotspots. And many of the plants are threatened, and many of them are extinct because of the anthropogenic activities and the construction purposes and many of the civilization activities. Friends, the term biotechnology is 50 decades old, but the technique is very ancient. The application of biotechnology is it is the science it's the application of science and technology to the living organism, as well as the parts of parts, the products and models. That is the technology used to develop various kind of the products by using the microorganisms. Here, many of the plants and also the microorganisms. So many organisms are exploited in industry for the production of the valuable kind of the products. So the impact of biotechnology on the medicinal herb management, that is the validation of the traditional medicines, that is the identification, classification and documentation of the, and then the identification of the authentic drug material. Because as you know, that is the, when you study the classification, that is the artificial system of classification, natural system of classification, and the phylogenetic system of classification. For artificial system of classification, starting from Theophrastus, the father of botany, and then the linear system of classification, and many of the system of classification. Even to today, for its practical purpose, we are following that system. Then phylogenetic system of classification, Arthur Cronquist system of classification. But even till today, it's very difficult to identify many of the plants because of the similar morphological structures, but they belong to the different species. That's why the many of the parameters are used, that is the DNA studies and also the phytochemical studies. For example, the flavonoids. The flavonoids are also used as the one of the chemical tool to in the classification, in the identification of the plants. Different kind of the flavonoids are present in different species of the plants. But there is a slight difference in their prosthetic groups. That is called as the chemotaxonomy. So that is the genomic studies and also the chemotaxonomy is used in the classification of the plants. Then identification of the authentic drug materials. Nowadays, many of the adulterant drugs are mixed with the genuine drugs. In the international market, it is rejected because of the presence of the adulterant materials. As you know, the pepper in the that is the papaya seeds are mixed with pepper, and even Emidasmus indicus, that is the Indian sarsaparilla. For that, many of the roots are mixed, and the Withania somnifera roots. Many of the Solanaceae plants are roots are mixed with Withania somnifera. So that's why the authentic drug materials has to be identified by using the biotechnical tools. That is the isolation of the DNA, and a barcode has to be developed for the genuine drugs. Based on the barcode, we can easily identify the genuine drug materials. And then bioprospecting of the medicinal plant, that is the ex-situ conservation of the threatened species, because many of the plant materials are threatened because of the over exploitation, even for the medicinal purpose also. In such a condition, in situ and ex-situ conservation technique has to be followed in the conservation of the medicinal plants. And then isolation and characterization of the novel drugs to combat the diseases. The authentic drug material has to be identified from the material from the medicinal plants. Then that drug material has to be chemically synthesized to control the depletion of the population in the nature. Then cell immobilization and enhance production of the medicinally active constituents. So we can easily culture produce the active ingredients from the cell suspension culture, so that you cannot depend upon the natural population. The depletion of the natural population will be avoided. You can get the active medicinal constituent from the cultured cells. Then genetic improvement of the microbes for the production of the novel compounds. So many of the microorganisms are growing as an endophyte. They are subjected to stress in that condition. You can isolate the endophyte from the medicinal plants and culture the that endophytes, and from that endophytes, many of the active ingredients, that is the medicinal plants that are present, components that are present in the plants are isolated. For example, the best example is the Taxol. Taxol is obtained from Taxus baccata, a gymnospermous plant. Taxol is an anti-cancerous drug. That Taxol is nowadays obtained from the culture of the endophytes. From the endophytes, that is by culturing the fungi, you can able to produce the that is the active medicinal ingredients. Then screening of the therapeutic potency of the medicinal plants. So in order to confirm the therapeutic potency of the medicinal plants, the extract and also the active constituents, bioactive constituents, has to subject it to various kinds of the in vitro and in vivo clinical studies. Then only the therapeutic efficacy of the plants can be confirmed. Now, direct we'll go to the ex-situ conservation of the medicinal plants. Here, the some of the plants are showing, for example, Boswellia serrata and Taxus baccata, the Chlorophytum borealis, Berberis aristata, Aconitum, and also Podophyllum and Picrorhiza. These are some of the endangered medicinal plant. They are at the verge of the extinction. So the conservation of these endangered plant species is very important, because when Mahatma Gandhi visited the Sandur Hill Ranges, Ballari, he called Sandur Hill Ranges as the heaven of the India, where many of the endemic plants are that is grown in that area. But nowadays, many of the plants, like for example, Philanthus Narayan Swami and Crotalaria microphylla and Crotalaria sanduriensis, these plants are extinct. They are not available. So because of the over exploitation, that is the removal of this soil, and also the removal of the that is depletion of the plants, and as a result, many of the plants become crossed the threshold of extinction, and they become extinct. So the conservation of these endangered plant species by employing in situ and ex situ conservation method is very important now. Friends, only a few plants are subjected to rigorous phytochemical and pharmacological investigation, but many of the plants are yet to be investigation. Hence, a careful and extensive study of the medicinal plant is essential to identify new products. For example, here in ex-situ conservation, the tissue culture is commonly used. Culture technique is commonly used in the conservation of the medicine plants. Hereby, the plant cell, tissue, and organs are grown under the control condition. That is in vitro. It is also called as in vitro culture. In vitro means within the glass. Within the glass or within a culture conserved area, we can able to multiply and produce large number of the plant species. So tissue culture offers a quick and efficient method to exploit the medicinal plants meaningfully to meet the demands of the pharmaceutical industries and to reduce the in situ harvesting pressure from the natural forest resources. So this technology can be used to get more plantlets without destroying the natural populations. It is nothing but a classified field of biotechnology. It is an art of biotechnology. Here, literally, as you know, literally means within the glass. Here, the plant species can be propagated all around the year. Whereas the breeder, in conventional methods, the breeder has to depend upon the season. And many of the plants bear the flowers once in 2 years, once in 12 years, or once in 100 years, like that. So, for example, palm, that is palmera palm, that is even that is bamboo, it blossoms once in 12 years. But by using this technology, the plant species can be propagated through all around the year, and the breeder is no longer restricted on the seasons in the production of the plantlets. So within a small area, within a glass, more than millions of the plants can be grown by using a small and even a microscopic cell within a short time. And we can study the physiology and growth morphogenesis of the isolated plant parts. So within by using this technique, it is also known to study what are the growth requirements necessary for the growth of the plants, including the crop plants, crop and medicinal plants. Then morphogenesis takes place in that plants. Yes, please. And the regenerates are easily transported across the international boundaries. For example, the plants are, many of the plants are endangered plants, and also very important plants are grown in the culture condition, and they can easily export to the other countries also, because it is easy to transport across the international boundaries when compared to the natural populations. So tissue culture also has advantages in the metabolic production when compared to intact plants. When compared to intact plant, the callus produced from the ex-plant, the part of the material, either it may be leaf, root, or any part of the that is a part of a group of the cells is called as the ex-plant. It is used to culture. And when the media are supplemented with the more concentration of the axins, in such a condition, the formation of the callus takes place. Generally, the secondary metabolites like the alkaloids, steroids, glycosides, flavonoids, and also the terpenes, they are produced in the plants when the plants are subjected to the stress condition. For example, the terpenoids and flavonoids, they are predominantly present in almost all the plant. But the production of the biosynthesis of the alkaloids takes place only in some of the families. For example, Solanaceae plant and also Asclepidaceae plants and Apocynaceae plants. In these plants, the production of the that is the alkaloid production takes place. Whereas in other species, in other families, some in Pandanaceae and also Lamiaceae, in these families, the alkaloid production is not noticed. So by culturing the callus, we can able to produce the required biochemicals, bioactive compounds from the within the culture condition. So without depleting the natural populations. So sometimes when compared to intact plant, the production of the alkaloids or some of the compounds takes place from the callus. And the rate of cell growth, the biosynthesis in culture initiated from a small amount of the plant material. Here we can produce culture only a small amount of the plant material for the production of the callus and also the large number of the plants. The cultures are maintained under controlled environmental and nutritional condition. So since you are capable to, that is the plants are cultured under a controlled environmental condition. We can also alter the metabolic pathway for the enhanced production of the bioactive compounds. And the suspension culture offers an effective mechanism of incorporating the precursor chemicals. The precursor chemical has to be incorporated into the culture. So the metabolic pathway has to be changed. The cells are trained or induced to produce the active ingredients in higher concentration. And here, a new route of biosynthesis of the metabolites can be produced from deviant mutant cell lines. It is also possible. Suppose some of the compounds that are not produced in the intact plant, that can be produced in the culture condition because it is exposed to a new kind of the stress in the culture condition. And some of the cells have the capacity for transformation of the specific substrate to more valuable products. So here, many of the industrial products are also obtained by culturing the cells in the controlled condition. Here, the totipotency, that is, it is the capacity or ability or the potentiality of the cell to produce new kind of the cells or the tissues and the organisms. Here, it is a meristematic cell subjected to the differentiation. And from the differentiation, you can obtain the embryogenesis or organogenesis or cytodifferentiations. And here, the somatic embryos are also produced. It is called as embryos or the embryoids. And regarding the organogenesis, you can obtain the collagenesis, that is the differentiation of the shoots, rhizogenesis, differentiation of the roots, and collorhizogenesis, both differentiation of the shoot and roots takes place from the culture. When you trace the history, here, Haberland is considered as the father of the plant tissue culture. He was the first person to culture the isolated plant cells in vitro on artificial media and used the willow twig, that is the Salis Raxburghi twigs, and cultured on the Knopf solution, where he obtained the growth of the shoots, and he considered as the father of the plant tissue culture. Later, many scholars worked in this field. That is the totipotency, the potential or inherent capacity of the plant cell to develop into an entire plant. Or it implies that all the information necessary for the growth and reproduction of the organism is continued within the cell. And differentiation, it is the capacity of the mature cells to return to meristematic condition and development of a new growing plant, followed by redifferentiation, which is the ability to reorganize into a new organ. So here we have to study differentiation, that is the de-differentiation, redifferentiation, cytodifferentiation, and competency, that is the endogenous potential of a given cell or tissue to develop into develop in a particular way. These parameters has to be fundamental parameter has to be studied is the ability of the plants. As I mentioned, the Gottlieb Heberland, he was a German botanist, was considered as the father of the plant tissue culture, and it defined totipotency as the ability of a single cell to provide and produce all the differentiated cells in an organism, including extra embryonic tissues. Totipotentials formed during sexual and asexual reproduction include the spores and the zygotes. Then Gottred, he was the first person cultured the cambial tissues of the isolated tissues of the carrot, and there he found the formation of the callus. And he was also provided the evidences for the cell theory, that is it was proposed by Slater and Swan. Then Philip R. White, he was the first to establish indefinite growth of the isolated roots of tumor tissues of the plants. And we nowadays we know the White's media, it's very popular media and used in the tissue culture. Then many of the investigators did the milestone work, that is the Murashige, and even to today for tissue culture purposes we are using the media formulation by the great scholar Murashige and Scoop in the year 1962. Even till today we are using the modified Murashige and Scoop medium in the culture of the plant cells. So what are the factors affecting the plant tissues under in culture condition? That is the growth of the media. Here the macronutrients, the micronutrients, hormones, and the carbon sources play an important role in the calogenesis, rhizogenesis of the plants, including the differentiation and de-differentiation of the plant tissue. Suppose if any one of the nutrients are changed in the media components, the morphology of the plant culture tissue may be varies. Suppose if you change the nitrates in higher concentration, sometimes the overproduction of the collagenesis takes place, or sometimes the differentiated cells organs undergoes de-differentiation, converted into the callus. Similarly, the environmental factors like light, temperature, photoperiod, sterility, and the media, all these factors play an important role. So the culture condition is very essential. You have to maintain the culture in aseptic condition. Then only you will get the plant. A suitable light and the optimal temperature of optimal photoperiod is very essential in the growth and multiplication of the plant cells in the in vitro condition. Then explant sources. Usually the explants are selected from the different meristematic tissues, that is the shoot tip or the flower, the inflorescence tip. Sometimes the different species show different responses in amenability to tissue culture. For example, the explants are taken from the shoot, leaf, and also the flower. If that explant culture inoculated onto the same media supplemented with optimal concentration of the growth regulators, then optimal concentration of the nutrient, the response may be varies depending upon the explant used. Because in any of the morphogenesis, the interaction of the exogenous hormones and also endogenous hormones takes place. If these hormones are interacted properly, then you will get the differentiation or the shoot formation, root formation, the callus formation, like that takes place. Yes, anything? Yes, please. Hello? Okay, okay, we'll continue. No problem. Some possibility. So, then what are the application of the plant tissue culture? That is the micropropagation techniques, diaplant production, protoplot fusions, genetic engineering. So in micropropagation, any of the plant materials, that is the leaf, shoot, or flower, or anything, any even a single cells, are cultured, and from the callus or directly from the explant, large number of the plants are produced. For example, if you culture a small piece of the shoot on a culture media, you will get many plantlets and even thousands of the plantlets from a single piece of the explant. So you can produce large number of the plants within a small area. And diaplant production, that is the microspore, that is the in anther culture also, in microspore culture, we can produce the diaplant production, where the haploids, from the haploid cells, the haploid callus or the haploid plantlets are produced, and it is diplodized by using the culture treatment. We will get a diaploid plant, and it's more bigger when compared to the normal haploid plants. Then protoplot fusion, that is somatic hybridization. Here the two protoplots are fused, will get an hybrid, that is somatic hybrid. And genetic engineering, by using genetic engineering techniques, many of the plants are altered, the genes are altered in the production of the valuable products or the high yields. But the word micropropagation was first used by Weber for the cultivated plants and that were propagated vegetatively. Only the plants are propagated vegetatively, they use the word micropropagation. But it is a technique of multiplication of genetically identical units, the clones. That's why in the micropropagated plant, they are all identical, whereas the plantlets derived from the callus may be identical or not. Whereas the plantlets derived from the that is through direct organogenesis, they may be identical. The principal method of micropropagation is the essentially involves the multiplication of the shoots from the either axillary buds or the formation of the adventitious roots or the adventitious roots, etc. So this significant advantage of micropropagation over conventional method. This is a way when compared to the conventional method, within a short time, within a short span of time, we can produce the large number of the plantlets. But there are many problems in the micropropagation. That is, the tissue culture is much talked about the superior means of the propagation of the plants guaranteed free from the pests and the diseases. Actually, by using the explants, even in the plant also, apical meristem and the root meristem, they are free from the diseases. But the major problem is the contamination. We have to maintain the aseptic condition throughout the culture condition, throughout the production. In any of the instances, if any of the foreign bodies enter into the culture, it will spoil the whole slot. Whole slot will be destroyed because of the contamination. So the major problem is the contamination. Another problem is the of the media. Suppose if the explant culture subjected to inoculated on the media, the nutritional component requirements on the hormonal requirement is not congenial for the growth and multiplication of the plant. In such a condition, exudation of the phenols takes place because of the oxidation. In such a condition, the antioxidants like the PVP, that is polyvinyl pyrrolidine, and also the ascorbic acid and also the activated charcoal is incorporated into the media to avoid the oxidation of the media. Then types of the micropropagation, that is the shoot culture, that is the shoot tip culture, then the floral meristem culture, multiple shoots from the seeds because the seeds are cultured and the seed germinated. After germination, the hypocotyl, cotyledon, and the radical part of the plants are seedlings are cultured on the media. We can get the multiple shoots. Instead of one shoot, we can get multiple shoots. Then nodal culture, the node of the plant is also, the stem is also cultured. For usually every buds, flowers arises in the axil of the leaves. So in a plant, usually two buds are arises from the nodes, because if, for example, if the leaf is opposite type, where if the phyllotax is opposite type or opposite decussate type, where two buds are arises from the node. But in nodal culture, you can get more than two buds. Sometimes 10 or 15 buds are arises from the nodes. So if that buds grows into the shoots, then you can induce the if that shoots are transferred to the rooting media, you can get the intact plantlet. Then direct shoot regeneration from the explant. Sometimes the organogenesis of the roots, shoots, or sometimes the roots are also takes place directly from the explants without intervening the calla space. If the cytokinin concentration is interacted with the endogenous growth regulator, then the organogenesis of the shoots takes place. Shoot directly takes place. For example, even in the nature also, the production of the shoots observed in from the leaf of the biofilm. I think many of you botanists, you know about the biofilm plant, and also in Discidium, you find the in Asclepiades member, you find the production of the roots into the pouches. So that is adventitious roots. Here also the same type of the adventitious roots or the shoots are produced from the explant without intervening the calla space. Sometimes direct embryogenesis. Here, normally in the zygotic embryos, the fusion of the male and female gametes. For example, imagine a polygonum embryo sac. It is a 7-celled 8-nucleated embryo, a polygonum embryo sac, where the the antipodals and also the micropolar region, the synergids are there. So one of the pollen tube enters through the synergid and it liberates the male gametes. One of the male gametes fuses with the egg cell and develops into the zygote, and another one fuses with the 2 nuclei of the secondary cell and develops into an embryo, that is a triploid cell, endosperm cell. But here, similar with the top, the zygotic embryo-like structures are produced from the explants, directly from the explant without intergoing the calla space. That is direct embryogenesis. They are also called as embryoids or embryo or embryoids. Then indirect shoot generation from the morphogenetic callus. The explant first organized into the callus mass. Suppose if it is cultured on auxin supplemented media, 2,4-D, or sometimes Na supplemented media, it organizes into the callus, and then if that callus transferred to the cytogenic supplemented media, cytokinin supplemented media, for example, BAP or kinetin or TDZ supplemented media, sometimes the combination of the kinetin, the auxins and cytokinins are also used. In such a condition, the callus organized into the shoots and also the roots. So here indirect organogenesis takes place. As you know, as a botanist, you know that when compared to the higher plants in the lower plant, there is pteridophytes are considered as the first vascular plants. Whereas in gymnosperms and also the angiosperms, the vascular system is well developed. When compared to the in Astraceae member, the Magnoliaceae, even the Michaelia champaka is a big tree, and the Astraceae member, a small Triadax plant, it is evolved when it is more evolved when compared to the Magnoliaceae member, because where tracheids are there. Here, true vessels are there. Vessels are in direct contact. Xylem and phloems are well developed in Astraceae member. Whereas in case of the Magnoliaceae and primitive members, Magnoliaceae members, sometimes the presence of the tracheids or sieve cells are observed. So similar such of conditions are observed in callus, where a differentiated cells organized into before the production of the organogenesis. A differentiated cells organized into trachid-like structures, sieve-like structure, where then it will organize and completely develop into a true vascular system. And then before the development of any organogenesis, it is modified into a nodular structure. Then the nodular structure transferred to a green photosynthetic structure, and from that organization of the shoots takes place. So the many number of the shoots are produced from the callus. So it is called as the indirect organogenesis. Whereas in direct organogenesis, without interfering the callus space, the organization of the shoots takes place. And another one is indirect embryogenesis. Sometimes the callus, if you culture the explant, for example, a leaf explant on a media, it transferred into the callus. And if the concentration of auxins are reduced, for example, 2,4-D, it is reduced to 0.25 or 0.5 condition, then the culture is transferred to the subjected to dark condition. In such a condition, the production of the embryos takes place from the callus. So embryogenesis takes place through the callus, whereas earlier embryogenesis takes place directly from the explants. And another one is the suspension culture. Here the callus is transferred to the liquid media, and cells are separated, and the single cells are cultured, and sometimes if the precursor chemicals are added to the suspension, then the cells are induced to produce the active metabolites, secondary metabolites into the media. So it is an advantage, yes. For example, in the production of the alkaloid, in order to change the specific pathway, tryptophan is added into the culture media, into the suspension culture. So if the bioactive constituent is an alkaloid, the higher production of the alkaloid takes place. You can get the alkaloid from the liquid media instead of from the intact callus cell. So there are many examples. I am showing many examples for example, the micropropagation. Here, the micropropagation of the teak plant, well-known teak plant, that is the Tectona grandis. Here the shoot tips are isolated and the shoot tips are cultured on the media. So then it is transferred to the rooting media. So the organization of the roots takes place from the micro shoots. So many number of the plantlets are used. By using a single piece of the explants, large number of the plants are produced all around the year without depleting the natural population. Similarly, the embryogenesis. So somatic embryos are also produced from the culture, for example, in the teak plant, and many number of the plants are produced. So here, from the callus, the callus is induced to produce the embryos. So if the embryos are transferred to the optimal media, it grows into a complete plantlet. These are some of the that is the in vitro that is transformation of the plantlets of the teak. So in our department also, the protocol is standardized for the mass multiplication of many of the threatened and vulnerable medicinal plants of the Western Ghats. For example, Silastris paniculatus, it is popularly known as Jyotismati. It belongs to family Silastraceae. And then Diosporus cardifolia epineaceae, Entera perseta mimosae, Embelia ribis myrcinaceae, it is popularly known as Vidanga or Basmug. Then Androgophis paniculata, it is called as Colmeg, it is belong to family Acanthaceae, and also Androgophis alata. Then Narvilia xylonica, Ranunculataceae, and then Vedelia calendulaceae, an Astraceae member. So it is called as the Alvigarg, similar to Eclipta alba, Bringraj. Then Bredilias scandens, Cisalpania bundosella, then Eralcium rigens, then Dionolix elata, and Semicarpus anacardia. These are some of the plants are that is they used in culture, and many of our scholars standardize the protocol for the mass multiplication of the plantlets by using the tissue culture technique. For example, this is Silastris paniculatus. One of my students, Dr. Maruti, worked on that, where the cotyledon plants are used. So the seedlings are subjected to germination, and the seedlings produces the hypocotyl and the cotyledons. The cotyledon parts are isolated, aseptically isolated, and transferred to media, and it produces the callus, and from the callus, the shoot generation takes place. If the micro shoots are transferred to the rhizogenic media, that is the rhizogenesis takes place, and the intact plantlet is produced, and it's popularly known as Jyotismati, and it consists of the chemical ingredient, that is silapanin. Usually the seeds from the seeds of the oil, in oil is alkali extract, it is called as Malkanguni oil. It is commonly used in massaging and as a pain reliever. And from the nodal explant also, instead of a single that instead of producing the two nodes, here multiple shoots are produced from the nodal explant, and here from the leaf explant the callus is produced, and from the callus the differentiation of the shoots are produced. The shoots are again transferred to liquid media, that is rhizogenic media. An intact plant is produced. Similarly, the anthers, the inflorescence explants, and also the anthers of the plants are isolated and aseptically transferred to the media. From the anther callus is produced, and haploid plant is also produced from the by culturing the anthers. Another one is the Entera perseta. It is popularly known as the in Canada Ganape, and the seed is the biggest among the angiosperm, and the fruit is the longest among the angiosperm. It grows more than 1.5 meter, the seeds. And one of my student, Dr. Vidya, now she's working as a professor in Alvax College. She worked on this plant. One of the important properties, that is the dormancy is more than 12 years in this plant, since the seed is very big. In such a condition, she standardized the technique for the breaking up the dormancy. So the seeds are subjected to the seeds are boiled, then it is treated with the sulfuric acid, then the different parts of the seeds, that is, it is excised, the seeds are excised and transferred to the media. So both direct organisms and indirect organisms obtained from this species, Entera perseta. It's also one of the red listed plant, Entera perseta. And from this plant, she isolated the saponin compound entamine and she screened the wound healing and also the hepatoprotective property of these plants. This is another one. Two of my scholars, that is Dr. Shankarmurthy and also Kumar Swami, worked on this. That is Embellia ribis. It's belong to family Myrcinaceae, and this plant, that is it is popular known as Vidanga or Basmak. In Sanskrit, it is called as the Basmaka, Vayu Vidanga in Canada, and in many of the Ayurvedic preparations, like Pipali, that is Piper longum, and Embellia also used as one of the main ingredient or a vehicle in treating the disease, because it is a good antioxidant. It has a good antioxidant property. And this Embellia ribis is also considered as one of the threatened species. Only it is very sparsely distributed in the Western Ghat areas. So Dr. Shankarmurthy worked on this plant, and he obtained that is the direct organisms from the leaf explant. Here the leaf explant is cultured on the media. From the leaf explants, the many shootlets are produced without intervening the callus space, and then it is if it is transferred to the rooting media, the production of the roots takes place. Here the the formation of the embryo-like structure from the callus. First, on 2,4-D media, the leaf explant transformed into the callus mass. Then, if it is subjected to the dark condition and cultured at low concentration of 2,4-D, the production of the embryo-like structures are produced. They are called as somatic embryos or the embryoids. And these embryoids exhibit the similar structures like the globular, torpedo, and chordate shaped structure, similar with that of the zygotic embryos. And these embryos, when it transferred to the oxygen supplemented media, they develop into a complete plantlet. Here, from the leaf explant, earlier is the embryoid production. Now, here, it is transferred to the callus, and from the callus, even the differentiation of the shoots takes place. So, many number of the plantlets are produced within a small area. Even this is another plant, that is the Andrographis paniculata. That is, it is called as the colmate or Nelabevu. It is belong to Acanthaceae. Here, from the explant, from the leaf explant and all those stem explant, the formation of the callus takes place. If the callus is transferred to the cytokinin supplemented media, the differentiation of the shoots takes place, and also here the differentiation of the shoots and also differentiation of the roots. Many number of the roots are produced from the root callus. And from this plant, it consists of a flavonoid compound, that is the andrographulidone, and this compound is possess the hepatoprotective property, and the leaves are used to cure the hepatoprotective and also antioxidant property, whereas the roots are used as an anti-malarial drug, because within a plant the biosynthesis of the secondary metabolites, the biosynthesis of secondary metabolites varies. For example, if the metabolite synthesis in the leaves are not synthesized in the roots, because the stress condition is varied. This is another plant, that is the Bridilius candens. Here also the somatic embryogenesis takes place. So the leaf is cultured on an media, an MS media, supplemented with the growth regulators like the 2,4-D and NAA. The formation of the callus takes place, and from the callus, the organelles of the somatic embryos. So the somatic embryos are similar with that of the zygotic embryo with chordate and torpedo-shaped structure. Then it is developed into the complete plantlet. Here, this plant is also one of the very important plant. It's an anti-cancerous plant. From this plant, anti-cancerous constituents is extracted and it is screened. One of the important property in of the that is the culturing of the callus. So, if the callus is cultured on the same media with different nutrient formulations and with different growth regulator formulations, the morphology and the texture of the callus varies. For example, if the callus of the Bridilius candens, it transferred to 2,4-D and kinetic supplemented media, it is the color pinkish, the callus is turned to pinkish color because of the biosynthesis of the more amount of the flavonoid compounds. Here the flavonoids are synthesized, that's why it is the anthocyanin synthesis is more, and that's why it is a pinkish structure. Whereas if the same colors cultured on the different media supplemented with BAP, where it is transferred to the fleshy mass. Whereas on IBA supplemented media, if the same colors transferred to that IBA supplemented, it is converted into friable mass. Whereas the same colors, if it is transferred to the media supplemented with the Na and carnitine, where it is the brownish and with yellow whitish nodule or hard nodule or mass. So the texture and the morphology of the callus varies depending upon the type of the growth regulator used and the type of the nutrient formulation used. So for the expression of any kind of the morphological structure, the interaction of the endogenous growth regulators and also exogenous growth regulators play an important role.
Hello, sir.
Yes, please.
Yes, sir. Actually, time is up. So next class is at 11:40. We have to manage the time, sir. Actually.
Okay, 11:40 ga?
Yes, sir. Next class.
So no time ಇದೆಯಲ್ಲ 11:40 ವರೆಗೆ ಇದೆಯಲ್ಲ?
Sir, actually the time break ಕೊಡ್ಬೇಕಿತ್ತು participants ಗೆ. But it is okay. ಅವರು ಕಂಟಿನ್ಯೂ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ ಇನ್ನೊಂದು 5 minutes ಅಲ್ಲಿ ಸರ್ ಕ್ಲಾಸ್ ಇದು ಮಾಡಬೇಕಿತ್ತು ಸರ್.
Okay.
Okay, sir. Thank you.
So just I'll skip because we'll go to the other one. Here it is the differentiation of the shoots, roots, and also roots from the tissues of the Bridilius candens. And as you know, the Bacopa monera, it is popularly known as the Brahmi plant. It is belong to family. Anybody knows? Bacopa?
Yes, sir.
Yeah, which family?
Sir.
This is Scrofularia.
Scrofularia, exactly, sir. So here, this plant is commonly grown in the sewage areas, but one variety is called as Pragya Shakti, and this produces the higher concentration of the saponin compound, that is called as bacoside A, and that bacoside A used as a intelligence enhancer compound. That's why it is called as the bromine. Brahmi plant. Even the many Bajaj company used this plant for the extraction of the Brahmi, many kind of the Brahmi product, hair oils and other things. So here the somatic embryos are produced from the culture of the leaf explants, and from the somatic explants embryos, the many number of the plants are produced. And this is another one, Nervilia zeylanica. It is popular known as Agnibolli in Canada. And here it has a good antiviral property. Many of the viral diseases are cured by using this plant. So here also both the direct and indirect organogenesis is achieved from this plant. And this is the nodal culture of the Nervilia zeylanica. And this is callus culture. And this is one, as you know, Gedjuga, that is Caesalpinia bundosella. Here also from the cotyledon plants, that is the many number of the shoots are produced by culturing the cotyledon plants, and the plantlets are transferred to this one. And here also from the root explant, the root produces the callus, and then if it is cultured to oxygen supplemented media, and it is the growth of the shoots takes place from the root callus and the development of the plantlets. Here from the leaf plants, leaf explant, the organoids of the shoots. And this is another one, Heralcium. This is also one of the antiviral plant. Here also multiple shoots are produced. Then another part is induction of the somoclonal variants in the conservation of the plants. For example, as you know, the Gregor John Mendel, because in the year 1860, he laid the foundation for the modern biotechnology, Gregor John Mendel. So many of you know about the laws of the Mendel. So he worked on the pea plant. Here, by using the tissue culture technology, through the callus, the regenerants are produced. These regenerants show the variations when compared to the actual in vivo plant. The regenerated plants show different morphological structure. It shows to be the texture of the leaves, the size of the leaves, or varies when compared to normal plant. This is called as the somoclonal variants. Even in the Bredilia, Embilia also, the plantlets regenerated from the inflorescence culture. Here, the more than 100 plants are produced within a small test tube, but the morphology of the plant when compared to the leaf size and also the texture, the regenerants, the structure is entirely different because of the somoclonal variation. And this also studied by using the DNA of both the plants. Here, different band patterns are observed when compared to the normal in vivo plant, and also the plantlet regenerated from the inflorescence culture. So this is the dendrogram of that one. Even in the rice, for example, one of my students worked on the Karimundaga, the black rice. So here the callus regenerated plant, so different morphological structure. It is entirely different when compared to the normal, that is normal in vivo rice variety. Because of the somoclonal variation, the texture of the leaf, the size of the that is the fruits also very different. Can anybody know the fruit of the rice? Hello? Hello?
Yes, sir.
Caryopsis.
Huh? Yes,
Caryopsis.
How it differ from the akins? Akins and
Akins are
caryopsis.
produced in
Akins and caryopsis. So both the akins and the caryopsis.
Both of them have developed from superior ovaries, sir. Single unicarpellary superior ovaries,
Monocarpellary you say single
Superior
univalvular structure. But in case
Yes,
of
sir.
caryopsis, the wall of the fruit is fused with the wall of the seed coat. There is no difference. But in case of the akin, it is differ. That is a slight difference.
Yes, sir.
Yes, sir.
So here the this that is the various stages of the regeneration in the rice plant. And even the somoclonal variants also produced in banana also. Here, usually the banana is highly prone to the Panama disease. The Panama is caused by the pathogen Fusarium axisporum F. cubans. Even till today, many of the farmers are struggled to get the genetic crop material. Suppose in one field the Panama is infected, the whole area, entire area will be destroyed. Even on the next year also, if the farmers planted the material, healthy material, the disease will occur. But here, the disease-resistant clone developed against the Panama pathogen, especially the Musa paradisica, that is the even also Musa cuminata. For example, the Nanjanood rasbole is it is one of the geological indicator, but is highly prone to the Panama disease. Or most of the rasboles, even the Malnad rasbole, Nanjanood rasbole, and even the football, that is, it's very popular in central Karnataka. That where cultivars are highly prone to the Panama pathogen, that is Fusarium axisporum F. cubans. Here, the regenerates developed from the micropropagation is subjected to different stress condition where it is co-cultivated with the pathogens, and the media is incorporated with the resistant constituents. In such a condition, the plantlets grows, the regenerates grows along with the pathogen. That clones will be selected, so we can easily produce disease-free and also disease-resistant plant matter, because in the culture of the meristem culture, we can able to produce only the disease-free material. Whereas in co-cultivation method, we can able to produce disease-resistant clones. These resistant clones are resistant to this pathogen. So it is a kind of a variant. Here,